Hello students, I am back in the classroom again to continue our video lesson on Eukaryotic Gene Expression regulation. In my previous video, I was talking about chromatin remodeling complexes. So what are chromatin remodeling complexes? How they bind to acetylated histone tails. Once they bind, how they are going to slide and evict nucleosome to make space for TBP or other transcription factors to bind to core promoter. Especially Swicenate is the well-studied chromatin remodeling complex that can bind to acetylated histone tails and that can either slide or evict, or sometimes it can also change the conformation of nucleosome that makes target parts or core promoter accessible for TBP. That's how the transcription gets initiated. In this video, I am going to discuss about the technique we are going to use, whether any transcription factors or chromatin remodeling complexes or transcriptional activators or co-activators interact with DNA or not, or if they interact, where exactly they interact, what is the DNA sequence. That we are going to study in this video using ChIP assay. ChIP assay is nothing but Chromatin Immnoprecipitation Assay. So as I mentioned here, you can study the interaction between specific or modified proteins and genomic DNA. Right? You can study the interaction between specific or modified proteins with DNA, how those proteins are interacting or what kind of proteins are interacting with DNA. You can use ChIP assay to determine, right? And also you can study whether transcription factors or what kind of transcription factors are interacting with target gene. Because as I mentioned, eukaryotic RNA polymerase, it cannot recognize core promoter by its own. It requires transcription factor assistance from transcription factors. You can study using ChIP assay whether transcription factors, how they are going to interact with core promoter and what are the transcription factors assembled at core promoter, right? Again, importantly, you can also study the presence of histones with post-translational modification at specific sites. You know that H3K9, if it gets methylated, that forms heterochromatin, right? Same residue, if it gets acetylated, that makes euchromatin. You can all study that using H3K9 methyl specific antibody or H3K9 acetylated antibody. You will get that specific antibodies. Using those, you can study whether histone H3K9 is methylated or acetylated, right? So for this purpose, you are going to use ChIP assay, right? Although it looks very simple assay, but it's very hard to reproduce in the lab. Anyway, so this ChIP assay was first established by Gilmore and Liss in 1984. The assay was first established in 1984 by these two scientists, Gilmore and Liss. In that, they wanted to see association of RNA polymerase 2 and H4 with Drosophila HSP70 gene. They wanted to check how that RNA polymerase 2 and H4 can interact with the promoter region of HSP70 gene. So how did they study this technique? It's very simple as I mentioned, but when you do in the laboratory, it's very hard to reproduce. Okay, they grew cells with or without yield shot. Thing is here HSP70 can be HSP70 gene expression can be induced upon heat response, right? Heat shock. So that's why they grow Drosophila cells in the presence and absence of heat shock. Then treated with formaldehyde. Formaldehyde that cross-links DNA and protein. Because here, unlike immunoprecipitation, in immunoprecipitation what you are going to do, you just target any protein and then take antibody, right? And that antibody is coated with some agarose beads, protein A agarose beads, right? That protein A or protein G agarose beads can bind to specific antibody and then you can pull down the target protein. In the chromatin immunoprecipitation assay, you are targeting entire chromatin, right? You are going to pull down chromatin. What is chromatin? Chromatin is nothing but DNA and protein. You are going to use antibody against that protein, but you are going to pull down entire chromatin, right? DNA and protein. So they cross-link formaldehyde, then that cross-links DNA and protein, right? Then chromatin, you know that DNA is very big, right? You have to make that into smaller pieces, generally 100 to 400 base pairs. How you are going to make that? By sharing, sonication. You can use sonicator to break big DNA molecule into smaller one, or you can also use restriction enzyme. Generally, shearing is preferred because restriction enzyme, if you do not control, that chops everything, right? Then once the chromatin is digested to this length, right? This step you have to standardize. So how many pulse you have to use to make or to get the DNA chromatin size in this range. That is, you have to standardize, right? Then you are going to immunoprecipitate the cross-linked protein and DNA complex with specific antibodies. In their experiment, they used H4 antibodies and RNA polymerase II antibodies. Then once they used antibodies against H4 and RNA polymerase II, then they used protein A or G agarose beads antibody, protein A or G attached to agarose beads. Then that protein A or protein G specifically binds to IgG that we discussed in the last semester, third semester in immunology, right? So here, when you use antibody against this and then protein A, protein G attached to agarose beads, that protein A, protein G binds to the antibody. Then that agarose beads are insoluble. You can just centrifuge and then pull down entire the chromatin. Chromatin means that here H4 along with DNA, right? So then, if you want to see whether that protein or H4 RNA polymerase bound to that any DNA sequence. For that, you are going to cross links. You are going to reverse the cross link by heating. Right? They reverse the cross link by heating. Then they purify DNA and amplify and quantitate using quantitative real-time PCR. Because we know that genome sequence of Drosophila and other animals, right? Or flies. Based on that, we can design a primer, forward and reverse primer, and then we can amplify the purified DNA from chromatin immunoprecipitation assay. So here I am going to explain with a pictorial representation. First I will explain H4 and RNA polymerase II. Then I will take one more example for your convenience to understand how the ChIP assay works. Right? Here they breed cells with with or without heat shock for 1 hour, Drosophila cells, right, in petri dish. Then remove the medium, wash cells with cold PBS, right? Remove the medium, wash the cells with cold PBS, because cells are attached. You can just add PBS and then throw away the PBS and fix with formaldehyde, right? Once you fix, here I enlarge single cell, okay? So if you consider this one cell, inside the cell there is a nucleus, right? In nucleus you can see DNA, right? Along with the DNA you can see the heat shock. Once they provided or exposed the cells to heat shock, then obviously there will be HSP70 gene expression. To express HSP70, there are several transcriptional activators, co-activators are required, then RNA polymerase binds, right? So when you translate, you just imagine here one transcription co-activator CBP, crab binding protein attached to the DNA, right? Then H4 is here, right? Then RNA polymerase II. So they were interested in only H4 and RNA polymerase II. Then lies the cell, ChIP buffer, using ChIP buffer. The ChIP buffer that contains SDS and Triton X100 that can break the membrane. But this ChIP buffer does not, reverse the cross link between DNA and these proteins. Right? Next, isolate chromatin. Once you lyse the cell, then isolate the chromatin. Right? There are several protocols to isolate chromatin. Then shear or use restriction digestion. So once you digest the isolated chromatin, you can see these are the pieces of chromatin that are in the range of 100 to 400 base pairs. Right? Some fragments of DNA chromatin that contains RNA polymerase II, some they contain H4, and some they contain CBP, that crib binding protein. So now they were interested in RNA polymerase II and H4. What they are going to do now? So they distribute this chromatin. What is that? Digested chromatin into different test tubes. Here I can say use 3 epitaph tubes. In one epitaph tube, you take the chromatin, like some amount of chromatin, like 30 or 40 milliliter of chromatin, and then add RNA polymerase II antibody. Right? In second tube, you take an aliquot of the isolated chromatin and then add H4 antibody. Right? Here, whatever the fragments that are having RNA polymerase II, that bound to RNA polymerase antibody. Right? Again, here, whatever the DNA fragment that contains H4 will bind to H4 antibody. Here, in third test tube, you are going to add IgG as a control. It's a non-specific one, isotype. It is an isotype control because here RNA polymerase II antibody and H4 antibody, they are IgG class. Right? You want to see whether if antibody non-specifically binding or pulling down this. For that purpose, you are going to use IgG isotype. Right? So once you incubated with the antibody, right? Here I will immunoprecipitate using protein A or protein G with agarose. Protein A, protein G, these are all from bacteria, right? These are the dietary toxins. They are isolated from bacteria and they are attached to agarose beads. Right? Now agarose beads are insoluble. When you centrifuge after incubating with protein A, protein G, agarose beads, you can see here that yellow one is protein A with agarose beads that pull down the entire chromatin along with RNA polymerase II. Entire chromatin along with H4. Here, because antibody is not specific, just you added isotype, it doesn't bind to neither of these proteins. I just represented all three, whatever I shown here, CBP, RNA polymerase II and H4. Because antibody is just isotype control, it doesn't bind to any of them. Even when you use like protein G or protein A agarose beads, that binds to that IgG only. Still you can pull down, right? Here in this case, you are going to pull down chromatin with RNA polymerase II and RNA polymerase II antibody. Here chromatin along with H4 and H4 antibody agarose beads. But here you just pull down IgG and protein A G agarose beads because this antibody is an isotype control. It doesn't bind to any protein. Right? So then you are going to analyze the DNA sequence or you are going to amplify the DNA sequence. For that you have to reverse cross-link by heating and use proteinase K to digest the protein part. Here what we require is DNA, isn't it? So when you reverse cross-link and use proteinase K, by heating you are going to reverse cross-link. And then when you use proteinase K that chops all protein, you will get only DNA, not chromatin, here DNA, right? Now purify DNA and amplify using specific primers. Here in the case of RNA polymerase II, right? Or what is that? H4. We know that what is that? Here they targeted HSP70 gene, isn't it? HSP70 gene. HSP70 gene in Drosophila upon heat shock. We know the sequence of HSP70 gene promoter. In that promoter, the RNA polymerase II and H4 will bind, isn't it? So we know the sequence of HSP70 gene promoter. You just make forward and reverse primer and amplify using quantitative real-time PCR. Once you amplify using quantitative real-time, data will be like this. You can analyze the data like this. Here, RNA polymerase II. Here, these are RNA polymerase II and H4. So these two bars in the absence of heat shock, right? These two bars in the presence of heat shock. Here, the blue one is RNA polymerase II antibody, right? RNA polymerase II antibody. The white bar is IgG. Similarly, green bar is H4 antibody and white bar IgG. Here, without heat shock, even if you add RNA polymerase antibody, there is no enrichment. Enrichment means that there is no amplification of DNA, because here, when there is no heat shock, no requirement of HSP70 gene. When they are exposed to heat shock, you see here in the presence of RNA polymerase antibody, you can see the enrichment of HSP70 gene promoter. Right? That means here, upon heat shock, the RNA polymerase antibody pull down the chromatin, the DNA, that gets amplified by real-time PCR. IgG as a control, you did not see any amplification. Right? Similarly, H4, same result. Here, there is no enrichment. In the case of heat shock, H4 antibody pulled down chromatin along with H4. So that's why you amplified that DNA, you found enrichment. But IgG as a control antibody, you don't see any enrichment. I'll take another example. This is the experiment that Gilmour and this established, because they wanted to see the RNA polymerase II and H4 interaction at the promoter region of HSP70 upon heat shock. So I'll take another example here, general example. This cat8 is nothing but histonacetal transferase, right? It's a transcriptional coactivity. Cat8 means lysine acetyl transferase 8. HATS, right? Histonacetal transferase, but lysine acetyl transferase 8. So what is the importance of this cat8? So this cat8 plays very important role in embryonic stage. This has been proved in several animal models, but I did not see any human data on this. But this cat8 plays very important role in embryonic stage. Means the development of embryo in mouse embryo or person embryo, and they use a lot of animal models. This cat8 is very, very important for the expression of antioxidant genes. Here, peroxyredoxin 1, peroxyredoxin 2, and then glutathione peroxidase 1, glutathione peroxidase 2. So these antioxidant enzymes are very, very important in the proper development of embryo. So if there is no cat8 activation, these genes are not expressed. Remember, here this is histonacetal transferases. What it does, it is going to acetylate, right? Once it gets, one histone tails are acetylated, that CBP binds, chromatin remodeling complex comes and binds to that acetylated histone, and then it slides a repeat. This cat8 specifically acetylate H4 at K16. Okay? Cat8 specifically acetylate histone 4 at 16th position of lysine. That is very, very important for the expression of these antioxidant enzymes. In the first bar graph, same thing, enrichment. You see here, when they use cat8 antibody, right? So these are from mouse embryo cells. They isolated the chromatin and then enriched or amplified, right? For peroxyredoxin 1, 2 and glutathione peroxidase 1 and 2 promoter, gene promoter. Here, when they use cat8 antibody, you see there is enrichment. The red bar represents cat8 antibody chip, cat8 chip, and white bar with IgG. IgG is isotype. It's an isotype. There is no pull down of any DNA because it is non-specific antibody. Similarly, H4 K16 acetylated chip. They used specific antibody H4 K6 in acetylated antibody. That antibody specifically binds to this residue and pull down the chromatin. And then when they use that antibody, you can see that bar graph, that blue bar, all got elevated, amplified or enriched. In the case of IgG, there is no enrichment or amplification. So this suggests that Cat8 is important in oscillating H4K16, right? This H4K16 is very important to make data paths open. Remember, similarly, S3K9 oscillation is also important. But in this case, Cat8 specifically oscillate H4K16, that is important in the expression of all these antioxidant genes, right? So this completes ChIP assay. And then in my next video, I am going to talk about briefly about transcription factors, what are general transcription factors and what are sequence specific transcription factors, and enhancer, promoters, silencer, because you have all these in your syllabus. So in my next video, I am going to talk about that, and then after that we will see how that RNA polymerase 2 the general transcription factors play important role in the formation of the pre-initiation complex along with RNA polymerase 2. Then we can go to specific transcription factor like NF-kappa B activation, right? So that will complete most of the portions in the syllabus. Then remaining will be DNA binding motifs, and then eukaryotic gene expression regulation at the level of post-translational modification. That we are going to study ubiquitination, the role of ubiquitination on eukaryotic gene expression, right? Thank you.